Hey everyone, today we are going to look at metabolisms and enzymes. So enzymes are macromolecules. Their function is to speed up biochemical reactions, so they're catalysts. They're proteins, so we know that with proteins their structure is super important. So the tertiary structure has to be maintained so that the active site can keep its shape. So if you look here, the active site is where the substrate fits into the enzyme, and they're very specific, which takes into account that the substrate, there's only one substrate that fits in each enzyme, so they're specific, and that's sometimes called the lock and key model of enzymes. So if the hydrogen bonds are broken, remember in the secondary, tertiary, or quaternary structures, if those are disrupted, then this active site is a different shape, and so the substrate won't fit in and it won't work. So the shape of the enzyme is very important to the functioning of it. So the active site interacts with the substrate. So a couple things to know about enzymes is that they're named for their substrate and they always end in ase. So for example, sucrase breaks down sucrose. Lactase breaks down lactose. Reactions can either build and be anabolic, so they synthesize, for example, this one right here, or they can break down things and be catabolic, like in digestion. Some other things about enzymes is that reactions don't use up enzymes or alter them, so they're reusable, and enzymes are specific and only catalyze one type of reaction. Another idea with enzymes is what's called the induced fit model. So if you look at this picture, here's your active site, here's your substrate, and so the substrate fits in there and the enzyme actually squeezes around the substrate to make it a nice tight fit. And that's called the induced fit model. I always think of like when you put your shoe on, right? The place where you put your foot in is open bigger than your is an opening bigger than your foot, and so then when you tie up your shoes, it's kind of like what the enzyme does when it kind of crunches around it. So the substrate is almost complementary to the enzyme active site. So why do you think that the induced fit is important? So if you think about if the active site were the exact shape of the substrate, then that substrate would have to come in at exactly the right angle, and not as many substrates would be able to bind with the enzyme. So this just helps it be more accessible to the substrate. So another thing with enzymes are the types of reactions. So there are endergonic and exergonic reactions. So if you look at endergonic, here is the beginning of your reaction, right? And we'll talk about activation energy, but this endergonic reaction needs a lot of energy to get it going. And then the products have more energy than the reactants. So energy is stored in the products. With exergonic, your reactants start with more energy, a little bit of activation energy is needed, and then the products have less energy than the reactants, and so energy is released. So with endergonic, energy is put into the reaction and it's stored. With exergonic, it is released from the reaction. And then here is some different some words if you wanna write them down. So some examples of endergonic and exergonic reactions would be photosynthesis and cellular respiration. I would write down the equation. You need to know this and notice that photosynthesis goes this way and respiration, only difference is that it's going the other way. So for photosynthesis is endergonic because you get energy, solar energy coming in, and then glucose, which is the product, has more energy than carbon dioxide and water. If you're doing cellular respiration and going to the left, then energy is released as ATP and heat, and glucose has more energy than carbon dioxide and water, and so that is an exergonic reaction because energy is released. And over here, this is just another way to see. So carbon dioxide and water go in, energy goes in, you get a nice energy molecule, and then it gets broken down with respiration, and energy comes out. So enzymes affect the rate of biological reactions, and what they do is they lower the activation energy of an enzyme-mediated reaction, speeding up the reaction. So without an enzyme, there is this huge pump of energy that's needed to get this reaction going. Now with an enzyme, and then also this would be exergonic because the energy in the products is less than the energy in the reactants. So with an enzyme, if you notice here, the blue line is, the blue line is the activation energy required with an enzyme, and the red line is the activation energy required without an enzyme. So this is why enzymes speed up reactions, is because they don't, with enzymes working in the reaction, you don't need as much energy. So I would jot this graph down for sure with all of the different arrows. And the way the enzymes lower the activation energy of a reaction is that it orients the substrate. So you could have two substrates, and maybe when you're synthesizing them, one needs to be tilted, and so this is how they'll fit into the active site. And so it can orient the substrate so that they fit together better. They block out water, and what they can do is they can actually the enzyme can pull on the substrates, making it easier for them to go into what's called the transition state, which is the middle state before they become the products. So enzymes, like we've said, have a specific 3D structure, and you can kind of see that here. Here's your normal protein, and if the shape of that changes, then it's called denature. And so here is the shape of an enzyme that's been denatured, and the things that can do this are heat and change in pH. And what's happening, remember, to get it to this nice tertiary structure, quaternary structure, you have hydrogen bonds that are holding that together. So when the enzyme becomes denatured, those hydrogen bonds break. And remember, the primary structure of the enzyme or protein is intact, but the others disintegrate. So what does temperature actually do? So the optimal temperature of a reaction is where the reaction is going the fastest. And so when temperature is too hot, right, when you're over here, here is your rate of enzyme activity goes way down, because when temperature is too hot, remember those molecules or atoms are moving, right? So they're moving, and so if they're too hot, then those hydrogen bonds, which is an attraction, get broken. And so the enzyme denatures, the active site isn't functional anymore, it doesn't fit the substrate, and the reaction goes way down. So when temperature is too cold, so over here, the enzyme doesn't get denatured, but what's happening is that those molecules, the substrate and the enzyme, aren't moving around very much. They're cold, right? So the molecules aren't moving, and so they don't, there's not very many enzyme substrate collisions, and so that causes the reaction to slow. So cold temperature doesn't denature the enzyme, it just causes the enzyme and substrate not to hit each other as much, and so the reaction decreases. So pH is another thing that can denature an enzyme. And just so you guys have a little bit of background or reminder, pH is the measure of the hydrogen ions in a solution. So neutral is 7, right? Anything below 7 is acidic, and that is adding hydrogen ions. And anything that's above 7 is basic, and you're adding hydroxide or OH negative ions. So if you have a pH of 7, you have 10 times more hydrogen ions in a solution that has a pH of 6. And so this is a logarithmic scale, is how we talk about pH. And earthquakes actually are the same. So if you hear of an earthquake that is a 5.6 on the Richter scale, which is how they measure earthquakes, and then you hear of an earthquake that's 6.6 on that same scale, then that means that 6.6 earthquake was 10 times stronger than the 5.6. So there's also an optimal pH for enzyme-mediated reactions, right? And so there again, your rate of reaction and your optimum pH. And what happens is if you are outside of that optimal pH, then those hydrogen bonds, those hydrogen ions can disrupt the hydrogen bonds and again, change the shape of your enzyme and denature it. So concentrations of substrates and products also affect the rate of reaction. So in the beginning, an initial increase in the substrate is going to increase the reaction rate because you have more enzymes and substrate hitting each other, and so more reaction happening. Pretty soon, and that's this green line, pretty soon your substrate is going to be what's called saturated. So your enzyme is going to be filled with substrate, and so if you add more substrate, it's not going to matter because your enzyme can only work so fast to break that substrate down. If you increase concentration of the products, then what that means is that there's going to be less opportunity for the enzyme to hit a reactant, and so that'll actually slow the reaction down as well. So enzyme concentration, right, also affects the rate of reaction. If you have a greater concentration of enzyme, then your reaction is going to go faster because, again, there's more collisions with the substrate and the enzyme. If you have a lower concentration of enzyme, then you're going to have a slower reaction. So what are some other things that can change the reaction rate? Well, you can have inhibitors, and there's two kinds. The first one we're going to talk about is competitive inhibitors. So I think the picture would be great to draw. Actually, any of these graphs that are on this page or this PowerPoint would be great to draw. So this is your enzyme, right? Here's your active site. And what happens is a competitive inhibitor fits into that active site so that the substrate can't, right? So it's competing for that active site, which is why it's called a competitive inhibitor. They can bind reversibly or irreversibly. And like I said, if the competitive inhibitor is bonded to it, then the substrate can't bond to it. So if you have a high concentration of these competitive inhibitors, then the substrate can't bind to it and the reaction will slow. And sometimes the competitive inhibitor can be the product, which we'll talk about in just a minute. So non-competitive inhibitors, if you notice, it's non-competitive because, look, it is attaching to the enzyme in a site other than the active site. And this place right here, this region other than the active site, is called an allosteric site. So you can have allosteric inhibitors, and if you notice, what it's doing is it's actually changing the shape of the active site so that the substrate can't bind to it. And this also decreases the rate of reaction. This sometimes happens, like if you have a product that's being made, that product can fit into the enzyme and slow the reaction if you have too much product so that it is not in equilibrium. And then once the product gets broken down, then this would come out and then the substrate would be able to fit in. And that's it. So thanks, and hopefully you took good notes. You can back up if you need to, draw some more pictures, and we'll see you next time.